Welcome to our class today. This is our 7th lesson in topic inheritance and evolution for the IGCSE Year 10 biology class. Our subtopic today is genetic ratios and what we refer to as a test crosses. We'll be looking at how genetic crosses are done or test crosses, the ratios and the probabilities in which we are able to express the outcome of any genetical combination. So this is a continuation of a series of lessons we'll be dealing with. Previously we talked about the genotypes and the phenotypes that have been actually expressed by our genetic makeup. So by the end of this particular session, we should be able to express our genetic ratios and determine the genotype by the use of test crosses. Let's look at our lesson, what we refer to as the ratios, or the Youngman ratios, the offspring ratios. Now, these ratios are actually probabilities. The probability we're looking about a random chance that is likely to occur. So, for example, when a man marries a woman, there are 4 possible young ones, or the genotypic of the offspring, the head of the test cross. Or there are 4 possible outcomes, 4 possible combinations of their young ones. Now, this does not mean that they actually have 4 children. The fact of saying that there are 4 possible outcomes does not imply that they are going to have 4 children. It simply means that each time they have a child, these are the possible genotypes that may occur. So before we proceed with this, I would want us to watch a small clip on these genotypic crosses and see how they are likely to occur. Let's watch the clip.
A monohybrid cross is one which involves only one trait. For this example, we'll look at stem height in pea plants. And let's say that you were told that tall pea plants were dominant to dwarf pea plants. Typically, you'll represent the tall allele, therefore the dominant one, with an uppercase letter such as the uppercase T, and the dwarf with a lowercase letter because it's recessive, so we'll use a lowercase t. So let's say that you're asked to cross a heterozygous tall plant with a dwarf plant. Each plant needs to have a genotype, and the genotype is going to be made up of 2 alleles. In the case of the heterozygous tall plant, because it's heterozygous, it indicates the 2 alleles are different. For the dwarf plant, the 2 alleles would be the same, as that's the only way that you could get a recessive phenotype. After you've identified the genotypes, you need to show the gametes produced from each plant. In the case of the heterozygous, one gamete might have an uppercase T, another a lowercase t, but from the dwarf you would only get lowercase t gametes. The gametes are produced through meiosis and therefore have only one allele. From the gametes you can produce a Punnett grid, or sometimes known as a Punnett square, to show the probability of certain outcomes in terms of the genotypes of the offspring. So by adding together the alleles from the gametes of the parents, you can start to see the outcomes. First of all, the genotypic ratio would be 50% uppercase T, lowercase t, and 50% lowercase t, lowercase t. The uppercase T, lowercase t is obviously heterozygous, lowercase t, homozygous. For the phenotypic ratio, you have 50% tall plants that came from the uppercase T, lowercase t, and 50% dwarfed plants, and that came from the lowercase t, lowercase t.
That's a simple illustration of what you are dealing when you are talking about the outcomes or the ratios that you are calling the probabilities or the possible random chances. So in this case, we have a case whereby you are homozygous, heterozygous, tall, in the case of a dwarf. This one is a tall plant, and this one is a short or a dwarf variety. So if you cross these two, you would have the gametes separating, into T and small t, and other, these are the gametes. So such that this and that will combine to give us that case, this and that, another case of that, while this and this will combine to give us a small t, another t, and the other one t. So if you look at all these outcomes, they are 1, 2, 3, 4. So we are saying out of the 4 chances, you would have a case whereby you would have 2 individuals, 2 outcomes that are actually tall plants, whereas in the other case, we have 2 cases where you have short or the dwarf variety. So if this is actually 2 out of 4, which is actually a half chance, or better 50%. In other words, that is a half out of 100 possibilities, that will give you 50%. So that's what we are trying to, that's what saying in terms of probability and expressing the chances that are likely to occur. If you remember in the other case where we are dealing with the case of a medical problem, which we found was a cystic fibrosis, where we had a patient suffering from cystic fibrosis, where we had the normal case using capital F, okay, and other you may have homozygous. In the other case, if it's recessive, that. So if this is a case of normal, and this one is a case of a cystic fibrosis patient. So in a similar scenario, you can also be able to predict that. So in this case, there is a possibility. If this couple happens to marry, if this one is a man and this one is a woman, there's a possibility that 1 out of the 4 chances, the kings will have a genotypic arrangement of homozygous F, okay? And 1 out of the 4 chances that there is a possibility one of the kings could also have this actually 1 out of 4, and then it's also out of 4. So in that case, all you're saying is you can also have this chance, 1 out of the 4 chances, one being normal, homozygous F. That one can also be homozygous recessive F, meaning this will be a victim of that particular case. So there are 2 chances also in out of the 4 chances, or rather 1 out of 4, that out of this combination, you could have a child that is heterozygous. That means it's actually FF, okay? That is capital F, small f, this one, heterozygous condition, this homozygous, this is homo-recessive. So that means in this case, we have 2 out of 4. So the genotype of the offspring actually worked out only in terms of probabilities. So with small numbers in terms of population that are bigger with, they are unlikely to work out exactly. But with large numbers or a large proportion, the young ones from one cross, they are likely to be more accurate. That means if you carry out a test cross of more individuals, you are likely to have more accurate predictions. The larger the sample, the larger the population sample being dealt with, the better the results, or the more accurate the results that are likely to be obtained. That means that if this couple that had cystic fibrosis actually had, for example, 8 children, we might expect at least 6 out of the children to be normal, but 2 of them are likely to have a case of cystic fibrosis. But equally, they should not be surprised if they may have 3 children with cystic fibrosis. So this is a case of probability or ratios that are likely to occur. Now we now look at the test crosses. Now a test cross actually helps us to determine the genotypes. So an organism that shows a dominant characteristic could have either of the 2 possibilities or the 2 genotypes that are present. One, it could be homozygous for that particular dominant allele, or equally, it could also be homozygous for the dominant allele, or in other words, it could be heterozygous. So there are 2 two possibilities here. You could have an homozygous case and an heterozygous case. So if we can be able to find out the genotype of an individual with the dominant phenotype for a particular gene by crossing it with one which has an homozygous recessive genotype for the gene. If we do that, then this is what we refer to as a test cross. Just like in the previous example that you had been given an example, the video club that you just watched, we were given a genotype of tall plants and dwarf plants. So if you take the same, we know that tallness is dominant to dwarfness. So if somebody is tall, there are two possibilities. You could have the dominant allele T for tallness, or you could also have the same in that case. Now, this is homozygous. Homozygous tall. In that case, it is heterozygous. If the short variety, you would only have recessive. It is actually homozygous recessive. This one is for the dwarf variety. This is for the tall case. So that's to mean those two possibilities. So we can only determine the phenotype of a plant by crossing a tall plant with a recessive. We take a break. We'll be able to clear that debate in the next session.